Okay, so today's lecture is going to be mostly about metabolism, some of the pathways in metabolism. So we're going to start with glycolysis, and this is just a brief overview. I put this in here for you guys. We post all of the PowerPoints and all of the videos after the lecture, so you can always come back and look at anything that you missed. Okay, so quick introduction to cellular respiration. Humans oxidize glucose from start to finish through glycolysis, the Krebs cycle, and the electron transport chain. So the oxidation of glucose is the main form of energy that humans utilize, and oxidation is how we actually take energy out of the bonds from glucose. In the first two steps, not much ATP is produced directly. So in glycolysis and the Krebs cycle, we're not super concerned. They do produce ATP and GTP, but we're not super concerned with producing the ATP. We're actually concerned with creating the precursors and creating some of the molecules that will feed into the electron transport chain. So glycolysis produces 2 ATP molecules per glucose molecule, and the Krebs cycle also produces 2 ATP molecules per glucose molecule. So as glucose is oxidized throughout these processes, the electron carriers NAD+ and FAD are reduced to their high energy forms NADH and FADH2. So those are the two main molecules that I was just talking about that are super important for the electron transport chain. So really we can look at these first two steps, glycolysis and the Krebs cycle. We can really look, their main function is to create these high energy electron carriers. So anytime anything is reduced, it becomes higher energy. So this NAD+, it gains an H with an electron over there, and it becomes NADH. So it's a higher energy molecule than this NAD+, and therefore it's going to be able to donate those higher energy electrons in the electron transport chain, and it's going to power ATP synthase, but that is for another lecture. So glycolysis produces 2 NADH molecules per glucose. 2 spins of the Krebs cycle produces 6 NADH and 2 FADH2 molecules. So it's important to note that the Krebs cycle spins twice for every single glucose molecule. As we're going to talk about in a couple of minutes, by the time that singular glucose molecule makes it to the Krebs cycle, it is actually split up into two different molecules, 2 3-carbon chains. So these products, after glycolysis and the Krebs cycle, they feed into the electron transport chain. And in the ETC, 34 ATP are produced per original glucose molecule. So let's get into glycolysis. Glycolysis is highly conserved throughout all domains of life. And in fact, all living cells contain enzymes for the glycolytic pathway. So basically, all living cells are performing glycolysis as the main way that they produce energy. So glycolysis has been around for billions of years. The enzymes are present in all different cells. It's a super simple way to do it. Obviously, animals and humans have evolved even more efficient ways to create energy, like oxidative phosphorylation with the electron transport chain, but pretty much all living cells throughout all domains of life contain the enzymes for this glycolytic pathway, so they do perform glycolysis. And glycolysis is split into 10 steps, and we can even divide those 10 steps into 2 phases. So the first 5 steps are known as the preparatory phase, and the last 5 steps are known as the payoff phase. Sometimes the preparatory phase is known as the investment phase as well, because we're actually going to be spending ATP in order to prepare these molecules for the payoff phase, where we'll actually make some ATP and we'll make some of those high energy electron carriers as well. And it's also important to note that glycolysis takes place in the cytoplasm. As we move over to the Krebs cycle and the electron transport chain, we're actually going to move to a different part of the cell. And the cell does this specifically. It sequesters these different actions in different parts of the cell just because there are different enzymes present in different parts of the cell. So glycolysis takes place in the cytoplasm, but it might not necessarily be possible in the mitochondrial matrix just because there are so many other things going on in the mitochondrial matrix. And vice versa, maybe the electron transport chain or the Krebs cycle takes place by the mitochondrial matrix. It probably won't be able to happen in the cytoplasm because there are too many enzymes from other reactions that are, buzzing around the cytoplasm and interrupting. So everything has its place for a reason, and everything fits together kind of like a puzzle. So we're just going to go right into the steps of glycolysis. This is meant to be a brief review. I tried to put as much detail in it that I think is necessary for the MCAT. So glycolysis, there is a lot to memorize. I think it's a good idea probably to be very familiar with all the products and all of the enzymes. When we get to the Krebs cycle, I think the products are probably a little bit more important than the enzymes. But glycolysis, I think you should definitely be extremely familiar with and have a really good conceptual understanding of how it works. So we're going to dive right in. So the first step, a singular glucose molecule enters the glycolytic pathway when it is phosphorylated into G6P by hexokinase. So hexokinase is a type of enzyme. It's a kinase. And if you recall from other lectures, kinases, they phosphorylate is what they do. So they add phosphate groups to molecules. So hexokinase adds a phosphate molecule, to a glucose molecule. So G6P and hexokinase work on a negative feedback loop. An increase in G6P, which is the product of the reaction, will inhibit the activity of hexokinase, which is the active enzyme. So that's pretty simple to understand. We've been talking about negative feedback loops in a couple of our lectures. It's just one way that this specific part of the metabolic process is regulated. When G6P is upregulated, then hexokinase is inhibited. G6P is now unable to leave the cell, because of the negatively charged phosphate group. So obviously we have that hydrophobic phospholipid bilayer that's surrounding the cell. So now that there's that negatively charged phosphate group on there, G6P is no longer able to leave the cell, so it's sequestered. It has to stay in. G6P is also supplied to the glycolytic pathway by cleaving glycogen. So we have glycogen in our muscles as well. It's that sort of branched storage of glucose, and they're all sort of together. So when we cleave glucose molecules for glycogenolysis, they actually are released as G6P. So when you have G6P, it could be going to glycogen, it could be feeding into the glycolytic pathway. It could be a number of things. So if you look over to the left, I put this schematic. This is glucose. We see it requires an ATP over here, and hexokinase allows this glucose to turn into glucose 6-phosphate. So we look right over here on this 6 carbon, we have an additional phosphate group over here. So that's really the only difference between the glucose and G6P is this phosphate attached to this 6 carbon. And that's what hexokinase does. And again, it's important to note that this step is irreversible. So we have 3 irreversible steps in glycolysis. It's going to be step 1, step 3, and step 10. And we will go over that as we get to each individual step. But as of right now, I just want to make sure that you guys are following. We're just going to track this glucose molecule as it goes through glycolysis in the Krebs cycle. So we start with a regular glucose. We use up an ATP because remember, we're in the preparatory phase, the investment phase. So we're using that ATP and it makes a G6P molecule. And the only difference is that we attached right over here, we attach this phosphate group right up to the 6 carbon. Okay, so step 2, G6P is now converted into F6P. So we have glucose 6-phosphate. It is now converted into fructose 6-phosphate by phosphoglucose isomerase. This step converts the molecule into a usable form for the following steps. So step 2, we have G6P right over here. Phosphoglucose isomerase. It is an isomerase. It's the same exact molecule, just in a different conformation. You can look right over here. Fructose is a pentose. It turns it right into here, but the connectivity is basically the same. And that's all step 2 is doing. It is reversible. It's not like a committed step. It's not irreversible. It's just preparing the molecule for step 3. And step 3 is the really important one. So another phosphate group is added to the F6P molecule via the enzyme phosphofructokinase 1. It's often referred to as PFK1, and it produces a product known as fructose 1,6-bisphosphate. So this is an important step. You should definitely know phosphofructokinase, and you should definitely know fructose 1,6-bisphosphate. And like 1, it's irreversible. It uses up an ATP molecule because we're phosphorylating. And this is referred to as the committed step because this molecule can no longer be used in any other metabolic pathways like glycogenesis. So we saw step 1, hexokinase turned glucose into G6P. That's irreversible, but it's not considered a committed step because that G6P can still be used in other pathways. like we talked about, like glycogenesis. Once that molecule touches PFK1 and turns into fructose 1,6-bisphosphate, that is the committed step. This molecule is going to go down glycolysis, whether it likes it or not. And just a really quick note on nomenclature, this bisphosphate means that the 1 and the 6, bisphosphate means that the phosphates are attached to two separate molecules. If we had, say, a pyrophosphate, it would be two phosphates that are connected to each other, that are connected to one of the atoms. But the bisphosphate means that it's connected to two separate atoms. Yeah, so G6P turns into F6P down here, right at this diagram, and then phosphofructokinase, we're phosphorylating again. So we have right up here on this 6 carbon, we have that phosphate group, and then phosphofructokinase adds another phosphate group right on this 1 carbon over here. So it's important to just look at the slight differences, because each of these steps really is just a slight difference between the others. So it's decently easy to follow. It's just all of the byproducts and the final product that are so different from glucose that we need to pay attention to. So we now have fructose 1,6-bisphosphate, and we can move on to step 4. So in this step, fructose 1,6-bisphosphate is cleaved by fructose 1,6-bisphosphate aldolase, which sometimes is just referred to as aldolase. And it's cleaved into 2 3-carbon sugars, and these are known as glyceraldehyde 3-phosphate. It's known as GAP, sometimes G3P, but it's actually cleaved unevenly. So the 2 molecules that are created are not, they're not identical. So one of them is GAP, this G3P we just talked about, and the other one is called dihydroxyacetone phosphate, or it's just DHAP. So those are the 2 3-carbon chains that are produced from fructose 1,6-bisphosphate, and this step is reversible. The remainder of the glycolytic pathway, I guess it's good to note, is just going to be oxidizing 3-carbon sugars. So from now on, when we're talking about 1 glucose molecule, it's going to be 3 carbons. We're going to have 2 molecules of 3 carbons. So that's why before, when I was talking about the Krebs cycle, how we need 2 spins for 1 glucose molecule, it's because we now have 2 molecules, and both of them are 3 carbons. Which obviously add up to the 6 carbons of the original glucose that we had. So, like I said, it's a bit asymmetrical. So the gap that was produced can continue on to step 6. It actually skips step 5 because step 5 is just concerned with converting DHAP into GAP. Because DHAP we can't really do anything with. We need that gap in order to move on to step 6. So step 5 is here. We have DHAP that is converted to GAP, and the enzyme that's responsible for that is called triose phosphate isomerase. Again, it's just another isomerase. We're just switching some atoms around, but it's basically the same empirical formula. It's the same atoms. So after steps 4 and 5, 1 glucose molecule has been broken down into 2 molecules of GAP. After this triose phosphate isomerase works its magic, we have 2 molecules of GAP. And I thought it was also important to mention that at equilibrium, after this step 4, when we have GAP and DHAP, and then we try to convert DHAP into GAP, the equilibrium of that is actually 96% DHAP. So it's super favorable in the reverse reaction. When we're trying to convert DHAP into GAP, 96% of that is at equilibrium is going to stay as a product. So the reaction alone does not proceed forward very quickly. The reason why we're able to get so much GAP is because reaction 6 pulls it forward. So as soon as that GAP is created, it's going to be shuttled right onto reaction 6, and it's going to be pulled over to another product. So you have to sort of think about it like Le Chatelier's principle. If we have a bunch of stuff on the left of the equation, our products, and we have a very small amount on the right, when we take away from the reactants, when we take away from that small amount, we're going to start stressing the system, and it's going to shift over to the right. So it's going to start making more products. So we're actually pulling the products as they're being created because we're using them for other reactions. So that's pulling the reaction forward. And that's a common theme that you see throughout metabolism and throughout a lot of these sort of concerted systems, is that even if the specific reaction is not favorable, the reaction after it is probably very favorable. So it's going to be pulling it forward. All right, so we'll move on to step 6. Remember, our DHAP is now GAP, so we have 2 molecules of GAP right now, and this is now the beginning of the payoff phase. So we're finally going to start making some ATP. We're going to start making some of those high-energy electron carriers. And in this step, G3P hydrogenase, remember G3P is another name for GAP, catalyzes the oxidation of GAP to 1,3-bisphosphoglycerate. This is a reversible reaction, and this is the first step that actually produces the high-energy carrier NADH. So if we look up here, we're going step 6. We're converting, I think I actually cut off this product, but we're releasing an NADH, and the product is just going to, we're adding this phosphate group right over here. So we have a bisphosphate again. Remember, it means that 2 phosphates are connected to 2 different carbons. So after step 6, we have right over here 1,3-bisphosphoglycerate. And we also just produced that NADH. So that NADH is going to be super helpful when we get to the electron transport chain. Step 7, we have this 1,3-bisphosphoglycerate. And the high energy phosphate on the 1 carbon, that one that we just put here, it's going to be transferred to ADP to create ATP via phosphoglycerate kinase. So again, remember, we're in the payoff phase now, so we're not using ATP to hydrolyze, we're actually creating ATP. So we take an ADP right over here, we take off one of these phosphates, boom, we get an ATP, and we get our product over here, which is 3-phosphoglycerate. So now we have this glycerate with only 1 phosphorus, with only 1 phosphate group. Yeah, and those are the products for step 7. Okay, we then have step 8. So right over here in this diagram, we're starting over here. This is our 3-phosphoglycerate that we just produced in step 7. And just keep in mind that we're looking at these 3, they're looking at a 3-chain carbon right now. So for every singular glucose, all of these steps after it's split, they're all happening twice. So we're only going over it once, but for each individual glucose molecule, it happens twice because we have 2 3-chain carbons, 2 3-carbon chain molecules. So step 8, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So the mutase is just moving the group. The phosphoryl group on carbon 3 is removed, and a phosphoryl group is added to carbon 2, hence the name of the product molecule. This step is also reversible. So the only difference in step 8, we're just moving that 3, we're just moving that phosphoryl group from 3 to 2. That's really it. And then we go to step 9. The 2-phosphoglycerate is dehydrated into phosphoenol pyruvate, which is usually just abbreviated as PEP, and it's catalyzed by the enzyme known as enolase. And this reaction is also reversible. And PEP is a super important molecule. We look at it in a bunch of different metabolic pathways, just going to and from. You know, we look at it in gluconeogenesis as well, just the opposite way. But PEP can now enter the final step of glycolysis. So that brings us to step 10. Pyruvate kinase catalyzes the conversion of PEP into pyruvate by transferring its phosphoryl group to ADP to create an ATP. So the only difference between phosphoenol pyruvate and pyruvate is that we're removing that phosphoryl group. So we tack that phosphoryl group onto an ADP. And in step 10, we come out with an ATP and a pyruvate. So those are 2 products that we definitely want. Pyruvate is the final product of glycolysis, and it'll go through 1 more modification before it can hit the Krebs cycle. So the product pyruvate, immediately after step 10, is created in its enol form, but it quickly tautomerizes to the keto form because the enol form is super unstable. And the tautomerization pulls the whole reaction forward and renders step 10 irreversible once pyruvate is formed. So this is going back a little bit to what I was just talking about a couple of minutes ago. This, the original reaction from phosphoenol pyruvate to pyruvate is technically reversible, but because the enol form of pyruvate so quickly tautomerizes to the keto form, and the keto form is so much more stable than the enol form, it's not going to be able to flip back to the enol form, and it's not going to be able to reverse back into phosphoenol pyruvate. So that's why this step is considered irreversible. So I'm just going to repeat that one more time because it's kind of important. Phosphoenol pyruvate reacts with pyruvate kinase. It creates ATP and it creates pyruvate, which is in its enol form originally, immediately. Once it is created, that enol form very quickly tautomerizes. That's the word that we use for transforming between the two forms. It immediately goes to the keto form. And it's very hard to switch back from the keto form to the enol form because the keto form is so much more stable. So because it's having so much problems getting back to the enol form, it's not going to be able to do the reverse reaction and turn back into phosphoenol pyruvate. So because of that, we consider the entire step 10 irreversible. So once we create pyruvate, it's not going back to phosphoenol pyruvate without the help of another enzyme that we're not going to get into in this specific lecture. Yes, and as Janelle said in the chat box, the keto form is almost always more stable than the enol form. Yep. So the two pyruvate molecules formed by glycolysis will head to the pyruvate dehydrogenase complex to be converted into acetyl-CoA before the Krebs cycle. So acetyl-CoA is actually what's spinning the Krebs cycle. So before we get to the Krebs cycle, we have to go through this one more step, basically, which is known as the pyruvate dehydrogenase complex. And we call it a complex because it's actually one big thing that's made up of 3 different enzymes. So E1, E2, E3. I didn't really get too in depth with it. You don't really have to know the specific names. E1, E2, and E3 are really sufficient. So after one glucose molecule makes it through the glycolytic pathway, 2 pyruvate molecules head to the mitochondrial matrix to the pyruvate dehydrogenase complex. So like we were talking about before, they translocate. They're at a different location than in the cytoplasm where we held glycolysis. So they go over to the mitochondrial matrix, and that's where this complex is. So in the PDC, pyruvate undergoes oxidative decarboxylation. So decarboxylation, if you don't know, it's just the release of a carbon dioxide from a carboxyl group. A carbon is released from the 3-carbon sugar as CO2, and the remaining 2-carbon entity is attached to a CoA group, coenzyme A, to form acetyl-CoA. A molecule of NADH is also reduced to a molecule of NAD+ is also reduced to NADH. So PDC is crucially regulated by AMP levels or AMP levels. High AMP levels indicate low energy in the cell and therefore upregulate PDC activity to feed the Krebs cycle. So AMP is the result of when you take ATP and you remove a pyrophosphate. So pyrophosphate are 2 phosphate groups that are connected. So we have adenosine triphosphate. When we remove 2 of those phosphates, we now have adenosine monophosphate. And it's a pretty good, when we have high levels of adenosine monophosphate, it's a pretty good indicator that our ATP stores are pretty depleted. So when PDC notices that these AMP levels are high, they're going to upregulate pyruvate dehydrogenase because we need more acetyl-CoA to spin the Krebs cycle. So if we take a look at this diagram over here, pyruvate enters this first complex over here, and it releases CO2 because remember, we're decarboxylating it into a 2-carbon structure. TPP is the cofactor that this first pyruvate dehydrogenase uses, the first part of the complex. It then goes over to dihydrolipoyl transacetylase, and that's where the acetyl-CoA is created. So it takes that 2-carbon chain, it takes the CoASH, which is just an abbreviation for coenzyme A. It's coenzyme A, and it actually has a thiol group, this sulfur right over here, and that's pretty important especially in terms of thiolysis, which we'll talk about in a little bit. But the second E2, the second part of the complex, is where the acetyl-CoA is released. And then the third part of the complex is where we take NAD+, we add an extra H and an electron, and we get NADH+ and a proton. Okay. All right, we're going to end. Cool. So 20% said A, 60% said B, 20% said C, which is great because 60% of you got it right. So let's take a look at it. A molecule that started as glucose can no longer leave the glycolytic pathway after it has been acted on by which enzyme? So hexokinase, remember, acts on glucose in the first step. It is not the committed step. When it can no longer leave the glycolytic pathway, we're actually looking for the committed step. So G3P hydrogenase and phosphoglycerate kinase are not the committed step. G3P hydrogenase and phosphoglycerate kinase, at that time, the molecule is already committed. But we're looking at the specific enzyme that acts on the molecule that is the committed step, and that would be PFK1. So remember, it's that, that conversion into fructose 1,6-bisphosphate. And that's just basic like definition. Step 3, this is something you're just gonna have to memorize. Step 3 is the committed step, and it produces fructose 1,6-bisphosphate. Right there, yeah, so phosphofructokinase turns F6P into fructose 1,6-bisphosphate. So the step is irreversible, and it is the committed step. So once we have that 1,6-bisphosphate, we're not going anywhere.